So we spoke about the medically cross-linking, or I call modulating. But what is going to be the future is Sheetal, and we all speak about cross-linking on the lenticule. But what I feel is going to be the next wave in the next maybe 4 or 5 years is looking at modulating the genetic itself, causing the gene therapy. And we got the first approval to make the vectors, which we are going to do. And hopefully this year we'll do a human trial on doing the gene therapy. The others, this is live from the gene therapy lab now. I would let Harkat handle this with Pooja on what this whole thing is all about. It's a small presentation, but this is going to be the future. All the extras and all the cross-linking, I hope that one day we'll address with this. This is a gene therapy lab.
Right. So this is the Center for Gene Therapy. It's this is now approved for producing clinical grade vectors for human trials. This is the first of its kind in India. Of course, there's a few pharma companies who are also trying to come up with it, but this is the first one with approval for making recombinant AAV vectors. So this is a view to the class 100 clean room inside, and there's what you can see inside is that these are all instruments. So a class 100 means essentially there are about 20 particles in 1 cc of air, so which is about 9 logs lower than what you have outside in the air. So that's just to make sure that you ensure cleanliness and safety, so that the product made is suitable to be injected inside a patient. So now there's
Trilok.
somebody, yeah.
Can we have the live feed from the lab, please?
A live feed.
So essentially what we are trying to do here is that we spoke at length about the gene lysyl oxidase. So since we've studied the biology a fair bit, now it's time to harness that biology for an actual treatment. So Trilok, you can go in.
Okay.
So
no, this one.
This is sort of a view of the lab. You can see.
Yes.
That's Dr. Trilok. He's one of the experts who's producing the clinical grade vectors. He's wearing that spacesuit. So you have to go through a series of air changes, of dress changes, etc., to arrive at that final space. So this is, he has crossed over. This is the entry. So this is a space where you take a clean air and mist shower, and then all the particles are removed, and then you enter the next space where you will have additional air showers, etc. So what we are trying to point out here is that making the clinical grade vectors is a cumbersome task. For all the pharma companies, they would know how difficult it is to maintain factories where you have to have very low particle counts to produce the drugs, qualify
the camera
the
is
Indian
focusing
FDA.
elsewhere. He's in the mist shower.
Yeah.
That's why it's like that.
The shower.
Trilok, once you come out of the shower, you can bring the... Yes. They're able to see the door now. It's on an airlock, so it takes a few seconds for him to come out. Yes,
so
now he's out.
Fully automated, so you can't override it. So now he's entered. Now you can enter the airlock, the air shower.
So those black knobs are where you get the jet of air, which it's an additional...
So this is super clean air. So each of these areas that you have, these are maintained using separate air handling units, and each air handling unit has its own particle count that is measured. so that you achieve the cleanliness standards required by the FDA. So now you have entered the clean area. We have 3 areas. We have the main bulk production area, the cell culture area. Trilok, you can go in. So what you are seeing inside here are the bulk production area where the vectors are being made. So Trilok, you can show the other side. So here you have bioreactors. You can see these instruments inside the laminar flow hoods. These are bioreactors. So these bioreactors are used to produce the, so essentially these vectors are made in human cells. So these bioreactors are used to do that. And these are the controllers for it. And apart from this, we have 2 additional areas. Trilok, we can go to the other 2 areas, go to the ACTA. So the recombinant AAV vectors are one of the most expensive man-made materials in the world. So as you know, gene therapies that are approved, there's a recent one that's been approved for muscular dystrophy. That is about $3.5 million per dose. That's because it is extremely complicated to produce it, and there's a lot of IP that's around it. So this is a chromatography system that is used to purify these vectors, and then it goes to the fill finish area. So of course the big question is, why are we making a drug so expensive? So obviously it's not going to be that expensive. It's going to be at least 100 times cheaper in India when we produce it and give it to our patients. But over time, I'm hoping that the technology will advance even further, that we can give it for a much lesser price as well.
So we can stop the
Okay,
feed.
so thank you, Trilok.
I think you can tell what all diseases we are working on and then we show them.
Right, so apart from keratoconus, so keratoconus is going to be our first gene therapy human trial here in India. Obviously, it's super easy to do. You can give it intrastromally eventually. We have actually tested it in mice as an eye drop as well, and it works beautifully. But for initial trials in humans, we will be using intrastromal delivery. But apart from that, we are going to be, right after this, we are going to start our trials on Stargardt disease and most likely on dry AMD, on geographic atrophy. So we have the data done for it. Now,
so I think over the years we have worked extensively on lysyl oxidase, started from Dr. Rohit, Natasha, myself, and then now the senior most fellows like Anushri Ritika. So we've proven time and again that if lysyl oxidase is on the lower side, there is higher the risk of progression or deterioration of the disease. And there could be factors like inflammation which could be at play, but lysyl oxidase, once it degrades, even if you do a cross-linking, it may not work effectively, and which was published in this paper, that if your lysyl oxidase is on the lower side and if you do a cross-linking, it has higher risk of failure. So, and also there are reports that even if you do a transplant, there are risks of failure of transplant, and there could be a genetic factor which could be involved. So the question here was, we now have the proof, but can we have a gene therapy in corneal ectatic disorders? The therapeutics, yes, therapeutics are going to help, but that's going to help at an early level. In advanced level, when there is no lysyl oxidase or lesser, I think gene therapy would be the way forward. And so I will just, we have seen this.
Right, so this is what we were talking about. So this is a vector. This is an AAV vector. You can think of it as sort of a bag or a briefcase that is very efficient in delivering the gene into the cells, whatever your target cells are. In this case, we are trying to deliver it to the corneal keratocytes. So the reason AAV vectors are the gold standard around the world for gene therapy is because it's extremely safe. It doesn't cause any known human disease. And when you are making a recombinant AAV vector, you have removed all of the virus's genes. So the virus doesn't amplify inside, it simply delivers and that's it. The gene sits there and makes the correct form of your protein. So you get the therapeutic benefit.
So it's always a journey. So it started from animal model, and now we have gone to cadaveric eyes, and now once we have
Ex vivo
approval, we'll
human
be
models,
doing it on patients.
Yeah.
So just let's quickly go to the data. What we did was, one eye was injected with a control, which is here the GFP, and the other eye was, the same contralateral eye was injected with lysyl oxidase. And you can see when you do the gene expression analysis, just a quantification that how the lysyl oxidase levels increase in comparison to a control eye. And when your lysyl oxidase levels increase, because it holds your collagen into place, it's known. So that is going to increase the collagen 1 and 4 levels. But more importantly, what we have seen is MMP9, which is an inflammatory marker. We saw that the MMP9 was going down with lysyl oxidase therapy. So it was not only just enhancing the lysyl oxidase activity and the collagen levels, but it was also reducing the inflammation.
Yeah. So essentially, we have sort of a side effect of the therapy that it reduces the inflammation also. So that's an important discovery. We've now figured out why that is happening.
And then next we wanted to see what happens in ex vivo models. So we know that there are multiple uses of lenticules which we remove. So we took the same lenticules like from the CLEC surgeries, and we did the same therapy. One eye lenticule was treated with a control, and the other eye was treated with the lysyl oxidase. And what results you saw on the animal model, you could see the same being replicated in the human corneal tissues as well.
Right. And that's what you can see in the protein level, which is an immunostaining for lysyl oxidase. So you can see more lysyl oxidase in the LOX treated ones, lesser lysyl oxidase in the controls. And again, the same thing we saw in the animals with the eye drop. Here we see the same thing. Next.
Yes.
Right. So then Pooja and Shubhro, who did this work, they took donor human globes and injected the vectors in those globes. So now, if you look at the left, this is the control, and the right is your gene therapy, the AAV LOX gene therapy. You can see the stroma is more tightly arranged with their collagen fibers. So that is because the lysyl oxidase is cross-linking all of this. And this is using the same holotomography imaging that we had done previously, you had seen, so to show that the collagen density or the compactness is increasing. Go next. So the same thing, we mean collagen density increases, and we use something called hydroxyproline, which is sort of a remnant of collagen that is getting produced, and that you can measure, and with lysyl oxidase you have more of this collagen synthesis that is happening. Only one injection. So that's the beauty of this therapy. You do it once and that's it. You never have to do it. So the human clinical trial should start hopefully in the next few months. We have already received the facility approval. It's already CDSCO approved. Yeah. Yes, this is only for progressive keratoconus.
Advanced.
We are. So after we start the keratoconus one, then we will start the star garden. Yeah. Keratocytes are reduced in keratoconus. So one of the benefits of that we observed in case of lysyl oxidase is that it prevents cells from dying off. That's number one. In some cases, the keratocytes sort of transdifferentiate and have, there's this mild effect on multiplication as well. We are very careful about that, which is why we are going to do the dose escalation study in the human patients, because we don't want too many keratocytes getting activated either. But keratoconus is the opposite of wound healing, right? So in wound healing, you don't want keratocytes amplifying, you don't want them to lay down collagen, but in keratoconus, you want them to be activated and you want them to be producing more collagen. So lysyl oxidase is a biological molecule that causes both of those things and achieves that. So this is an atomic force microscopy. This is, so while we spoke about the collagen density increasing and the compactness increasing, we also have to show whether or not the elastic modulus improved, right? So that is the primary thing that you're looking for. So we used atomic force microscopy to measure the elastic modulus, and this is, these graphs are from goat eyes, and the next one is from human donor eyes. So these were eyes that were given the vector, and after 4 weeks, then we did the AFM analysis, and we find that we are able to increase the Young's modulus here.
So I think, so I think what future lies is in gene therapy. When
Yes, Dr. Marshall has already said,
Yes.
the future is in genes. So everybody has to get into that.
Can I ask, yes, knowing the brutal nature of regulators, what have you submitted as your index of success in terms of your trial? So you know what the FDA are like.
Right.
You have to have an index of efficacy.
Yes. So in our case, because obviously animals don't get any keratoconus, so we don't have any spontaneous models of keratoconus. So the data that we have submitted is from these sorts of structural analyses, where we have showed the Young's moduli getting improved. We have shown the structurally the compactness and everything else improve. What we are also submitting, of course, there's a whole bunch of safety data that we are not showing here. You know, your endothelium is okay, your epithelium is okay, etc., etc. We have induced keratoconus model, where we have put inflammation and eye rubbing together, and that causes the lysyl oxidase collagens to go down, your MMP9s to go up, and there is some amount of thinning. So we are using that.
Perhaps all I needed to know was, what can you measure in your patients, and the FDA says that's success, or at least efficacy?
Yes. So, so here we are going to measure MMP9 and lysyl oxidase in our tiers. And the patient selection is based on progression. So there has to be documented progression for at least 6 months, preferably to a year, in order for a patient to be enrolled. And again, of course, gene therapy is not going to be the first line treatment. This is if your Trelos or other things fail, is when you're going to use this.
Allows keratocytes to multiply?
Yeah. So when you have more of it, so there's a, yes, so lysyl oxidase, so you know that lysyl oxidase gets processed into at least 2 other forms, which are glycosylated, etc., right? So it's not us, but there's some labs working on retina who have shown that a certain proportion of the processed lysyl oxidase enters back into the cell and works like a transcription factor. So it works with other genes and works like a transcription factor. Now we know that it actually induces the SMAD pathway. So the SMAD genes are, the SMAD transcription factors are the ones that control extracellular matrix related genes, and they promote cell survival as well as amplification.
okay, thank you everyone.